Hey everybody, thank you for checking out this video. Guys, kindly support my work by subscribing to my YouTube channel, hit the notification button, and also like the videos and share the videos. And I want to say thank you to those people who have already subscribed and those who have been supporting me. Thank you very much once more, guys. In this video, I'm going to talk about DNA replication. DNA replication is a very simple topic. At the same time, it's very interesting. DNA replication. I thought of making this video, guys, because I know that on Tuesday, I don't know for sure if it's Tuesday or Wednesday, we're writing biology test 2, and also quiz. So I thought of making this video for the revision sake, guys. Okay? So in this video, let's talk about DNA replication. What is DNA replication? What do you understand by the term DNA replication? So DNA replication can be simply defined as the replicating of the double helix of the DNA, or we can say the process by which new copies of a DNA are formed. So the process by which new copies of a DNA are formed. Okay. So new copies of a DNA are formed, or we can say the replica of a DNA are formed. Okay. So that means that a copy of a DNA is formed. All right. So for a DNA to produce other DNAs, or for the double helix strand to manufacture other DNAs, it needs to replicate. All right. So there's going to be a parent DNA and also daughter DNAs. So these daughter DNAs, they come from the parent DNA. So the parent DNA should replicate. So now in this video, we are going to learn the mechanism by which this process occurs. So DNA replication, how does it occur? Is this very complicated? No, it's not. So imagine that you've got this DNA molecule, which is a double strand. Okay? Then you've got the nucleotides. All right? So these are the nucleotides. Okay? And then this DNA, it is running from 5' end to 3' end. Then the other one is running from 3' end to 5' end. Okay? So this strand is running from 5' end to 3' end. Then this one is running from 3' end to 5' end. Okay? So now, even on this one, we've got nucleotides. Alright, so nucleotides. Alright, so these are the nucleotides. Okay. So have you noticed something here? So me have noticed something. If you can see, this is 5, this point is 3'. Okay, this is 5', this is 3'. So there's a difference here. This one, it is going in this direction. Then this strand is going in this direction. Okay, or the numbers are decreasing in this direction. Okay, so for the first one, numbers are decreasing when you're going 5' to 3'. And then for this one, numbers are decreasing in the opposite direction. So this is a very important feature of DNA. This feature is one that causes complementarity between nucleotides. So this feature, it is called antiparallel. So we can say that the DNA is antiparallel, or the DNA strands, to be more precise, are antiparallel to each other. Okay? So what do they mean by antiparallel? When they say antiparallel, from the word parallel. That's where this word is coming from. Okay, so anti-parallel. So we know that parallel lines, they don't meet, right? Of course, the DNA strands don't meet. Okay, they form a helix or a twist, but they don't meet, or they don't cross each other. All right, so they are parallel, but anti, it means they move in opposite directions, or their ends, they are in opposite to each other, or they're opposite to each other. That's what I can say. Okay, so this one, 5' end to 3' end. This one is running from 5' end to 3' end. Okay, but in opposite direction. All right, so this is what we call anti-parallel. Okay, so the DNA molecule or the DNA strands, they're anti-parallel to each other. So it's a very important feature. So now, imagine that you've got, let us say that you've got this sequence, this is DNA sequence, 5' A, T, C, G, T, A, 3'. And then they tell you to find the complementary sequence, or the sequence on the complementary strand. How can you do it? So we know that 5', this 5' here, okay? So let us say this 5' this side, this 5' here, it is complementary to 3'. So what do they mean by complementary? They just mean that 5' partners with 3'. Okay? 5' goes with 3'. A goes with T, and then T goes with A. Just like that. C goes with G. G goes with C. Okay? So that is complementarity. So that's one of the best features of a DNA. Okay? So the DNA has a complementarity nature. So now we can say that the sequence on the other strand, it is 3'. Then A adenosine goes with what? Thymine, T. Then thymine goes with adenosine. What about cytosine? Cytosine goes with guanine. Then guanine goes with cytosine. Thymine goes with adenosine. Then adenosine goes with thymine. 3' end is complementary to what? Is complementary to 5' end. Okay. So when two things are complementary, it means that you can determine the other just using the other. Okay. So using this strand, we can determine the second strand. That is what complementarity is all about. Okay. So this is a great feature of the DNA, and you need to know that the DNA sequence, or the DNA sequences, or we can say the DNA strands, they are complementary to each other. All right. So now let us talk about the DNA replication. Let us now talk about the DNA replication detail. So DNA replication, it is described to be semi-conservative. Okay, so they say DNA replication, it is semi-conservative. What do they mean by semi-conservative? Okay, so you need to understand the prefix which is here, semi. What does semi mean? So semi means half. Semi means half. Okay, semicircle, it is half circle. Okay, then what about conservative? Conservative just means something that can be conserved, or something that can be kept for later use. Okay, so the DNA, it is partially, the other word for semi, it is partially. Okay, so we can say it is partially conservative. All right, so a partial part of the DNA can be kept for later use during replication. That's why we say DNA replication, it is what? DNA replication, it is semi-conservative. DNA replication is semi-conservative. So now let me try to demonstrate what I'm talking about. All right. So now imagine that you've got this strand, okay, of a DNA, and then you've got another strand. of a DNA. All right, forming a double-stranded DNA. All right. So when this strand replicates, so when this DNA replicates, or when this double strand replicates, it's going to form. So from this part, we are going to have a complementary. All right. So let me use another color. So we are going to have a complementary. We are going to have a complementary strand. So there's going to be a copy from this strand. We are going to get a copy from this strand. All right. So this is a copy using the DNA polymerase 3. Okay. So there's going to be a copy emerging from this part. All right. And then this let's call this one a parent strand, right? So this parent strand, it is going to be conserved, or to be part of the new daughter DNA. All right. So this is going to be here. Just use the same color. So it's going to be there. All right. So this one, the same way it is going to be part of the daughter. And then from this one, there's going to come. We're going to get another. a copy of this, which is this one. All right. So a copy has emerged from this, which is there, and then the same this one. It has gone there, forming a double helix strand. All right. And then what about this one? This one is also going to form another DNA molecule. So this one will be also conserved, or will be also used. Then from this one, the same one, this one, another one or another copy is going to be formed. So let us say, let's give it, all right, let me give it blue to differentiate. All right. So this is a copy. All right. So this is a copy of this, and then this is a copy of that. Then this one just come here. So let us say this is a parent strand, and then this is a new strand, and then even this one, new strand. This one, parent strand, because it's from the parent DNA. This one from the parent DNA. Okay. So it's like only one out of two strands, only one new strand is formed, and then the other strand comes from the parental DNA. All right. Yeah. So in each and every newly formed DNA, there is one new strand and one parental DNA strand. All right. Forming double strands, forming two strands. So that's what they mean by the term semi-conservative. Okay. So it is partially conservative, or this DNA, half of it goes there, which is semi, and then the other half goes there, semi-conservative or semi-kept. All right. So this is what they mean by the term semi-conservative. So the DNA replication is what? Semi-conservative. You have to understand that and know that it is very important. All right. So now let us see what happens during replication. So let us say if you've got, this is a DNA molecule. Okay, DNA molecule. This is a DNA molecule. Okay. All right. So this is a DNA molecule. All right. Okay. So now, if you've got, let us just label this part as 3' end. All right. So okay, I'm just saying this is the 5' end. Then here it is 3' end. Okay. So the yellow line starting from 3' end to 5' end. Then the white line, or the white strand, we say it is running from 5' end to 3' end. Okay. So what you have to understand here is that DNA replication occurs from 5' end to 3' end. All right. Or the nucleotides are added from 5' end to 3' end direction. Okay. So how does DNA replication occur? So the first thing for DNA replication to occur, we've got what we call the primer. Okay? So what is a primer? So a primer is just a short sequence of RNA molecule. So a short sequence of RNA molecule which initiates the DNA replication. It's what we call DNA. What we mean the primer. All right. So this is what we call primer. A primer simply means a short sequence of DNA of RNA which initiates DNA replications. All right. So the primer will first come and attach at the origin of replication. So this point where replication starts, so at the point where replication begins, it is called origin of replication. So the DNA primer will come and attach at that point. So when it attaches at that point, what's going to happen? So there's going to be an enzyme called DNA polymerase 3. DNA polymerase. All right. Polymerase 3. Okay. So DNA polymerase 3 is able to recognize the primer. So it's going to come. It will come. When it comes, it's going to recognize that primer there, or that short sequence of mRNA. Okay. So when it attaches there at the primer, it will start moving. Okay. So this primer, it is formed by an enzyme called what? Primerase. Okay. So now let us say that we've got a DNA molecule which is about to start replicating. So this DNA molecule is made up of two strands. So these two strands, they are anti-parallel to each other. Okay? So which means they've got opposite orientation. Okay. So the first strand, let us say it is running from 3' to 5'. Then the complementary strand is from 5' to 3'. Okay? So the direction 3' to 5' is called upstream. Then 5' to 3' is called downstream. So this one downstream. Then the other one upstream. Alright. So DNA replication starts with the primer. Okay? So what is the primer? So a primer is just a short sequence of RNA. So it's about 10 to 15 nucleotides. Okay? So just a short sequence of RNA which attaches where? At the origin of replication. So the site where replication begins, it is called origin of replication. Alright. So let us say that this one, it goes and attach there. Alright? Okay. So mostly, and I can say always, especially in eukaryotes, the primer or the origin of replication is found where the density or where there's high concentration of AT. Okay? So when there's high concentration of AT bases, that's where the origin of replication is found. What is the reason behind that? Why? Why does that the replication begins where there is high density of these two nucleotides? The reason is simple. Between adenine and the thymine, there are double bonds. But between guanine and this cytosine, there are triple bonds. And you know that it takes less energy to break double bonds than breaking triple bonds. That's the reason that DNA replication, or the origin of replications, is found where there is high density of A and T nucleotides, bases. Okay? So when there's high density of A and T bases, that's where the origin of replication is mostly found. Because it's going to be easier for the DNA polymerase 3 to break these two bonds then breaking 3 bonds. Okay, so after the primer recognizes, or after it finds the origin of replication, or where there's high density of A and T bases, then an enzyme called DNA polymerase 3. So DNA polymerase 3 will come in. So let me just say DNA polymerase 3 is going to come in. So it's going to come in and attach there at the primer. So when it attaches there at the primer, it will start adding nucleotides in the 5 prime to 3 prime direction. Okay, that's the reason we say DNA replications occurs in the 5 to 3 prime direction. Okay, in the 5 prime to 3 prime direction. All right, so the DNA strand is 3 prime to 5 prime. Then DNA replication is going to be the opposite of that, the complementary of that, because we're talking about the copy here. All right, so the copy of this DNA strand. Okay, so it will start with 5 prime and then end with 3 prime. That's a copy. All right, so it's very important. You should know that DNA replication occurs in the 5 prime to 3 prime direction. Okay, so now let us say that we've got. All right, so we'll say this is the double helix. Okay. Okay, so this is a double helix. Okay. So when the DNA polymerase recognizes the origin of replication, all right. So when the DNA polymerase 3 recognizes the origin of replication, let us say it is there, there's going to come in an enzyme called helicase. So what does helicase do? So helicase, it unwinds or it uncoils what? The DNA molecule. So it's going to unwind the DNA molecule. So this enzyme is called helicase. Okay? So the function of helicase is to unwind this DNA molecule. So it's going to unwind, then separate them. All right? So it unwinds, it's going to break the bonds between what? It's going to break the bonds between the bases. All right? So between these bases, or the bonds between nucleotides, it's going to break them. All right. So this DNA, or this helicase, it starts from where? From the origin of replication. Because imagine that earlier, the DNA was like this. All right. The DNA was like this, okay? Was just like this. Then here we have got bonds here. You have bonds, okay? Those are hydrogen bonds. So first, it's going to break these bonds. Then this will be like this. So when these bonds are broken, so the enzyme helicase is going to be breaking bonds like that. All right. So it's going to start breaking the bonds or separating these 2 strands from the site called origin of replication, then going in this direction. All right. So we can say that this is the 3 prime to 5 prime end. Then this is 3 prime 5 prime end. All right. And we said that DNA replication occurs in the what? In the 5 to 3 direction. 5 prime to 3 prime direction. Okay. So when this DNA polymerase 3, remember we have got 3 types of DNA polymerase. We've got DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. So DNA polymerase 3, it is used to replicate or to add nucleotides. All right, that's what I can say. That's a better way. It is used to add nucleotides, okay, to the replicating strand. So it is going to start from there at the origin of replication, okay, where there is the primer, and then start moving in this direction. okay, in the 3' to 5' direction. Then it's synthesizing what? Going to be producing or copying the DNA strand in the 5' to 3'. It is moving the 3' to 5'. Then it's producing a copy of a DNA in the 5' to 3' direction. So you have to understand. So it will be going like that. And then these Y-shaped strands, they are called, these two Y-shaped, they are called the replication fork. So because it's looking like a fork, okay? So when you look at it, it's looking like a fork. So this is called the replication fork. All right. And then when you see, you can see that there is a bulge there and a bulge there. So this one, it is called replication bubble. It's very interesting and very fun. So it's a replication bubble, and I'm sure it's very easy to remember. So replication bubble. Okay. So this Y-shaped there and that, these two strands which are forming like a Y shape, okay? They're called replication fork. Okay? And then this part, where there's a bulge, this part, okay, this part and this part, this is called replication bubble. All right, the whole thing, okay. So now, as the DNA polymerase 3, it is moving in the direction of 3' to 5'. We've got another part here. We've got another strand this side. Okay, and when you look at this strand, it is running from 3' to 5' in the opposite direction. So what happens to this strand? Is it just going to be dormant, not doing anything? No. Even on this strand, there's going to be replication occurring. So the replication is going to be there, even on this strand. All right. So now let us see what happens on the other strand. So our diagram was, we say, this is the diagram. This is a DNA. All right. So this is the DNA. Then we've got the DNA polymerase 3 moving in this direction. Then we've got, this is the 5' end, this is the 3' end, and then here we've got 3' end, 5' end. All right. So this DNA is moving in the 3' end to 5' end, producing a continuous strand. So this one, or this strand, 3' end to 5' end, it produces a continuous what? Continuous strand of DNA. All right. So it's going to produce a continuous strand. Then this one, what's going to happen to the other strand? So there is going to come in an enzyme. Or in other words, I can say, let me just say, a primer. So a primer is going to come there. So this primer will attach there. In eukaryotes, we can have a multiple origin of replications. In prokaryotes, we can only have one origin of replications. But when we talk about eukaryotes, for example, we talk about cells in human beings, cells in animals, we can have many origins of replications. Okay, so there we can have an origin of replications, a primer attached there, and you can have another origin of replication. Okay, so this one, this strand here will be producing a continuous strand in the 5' to 3' direction. Then this one, there is going to produce a strand up to there, and even from there, it's going to produce another strand up to somewhere. Up to somewhere here. Okay, so you're going to have a strand here, then a gap between another strand there. Okay, so we can say that this one, this strand, which runs from 5' to 3', it produces a discontinuous. So it produces discontinuous strands, while the other one produces continuous strands. All right. So this one is producing continuous strand, this one producing discontinuous strand. So we can say DNA replication is semi-discontinuous. Semi-discontinuous or semi-continuous, whatever you choose. Okay? it is partially, because half it is continuous, then the other half discontinuous. So you can say it is semi or it is partially. Semi just means partial or half. Okay, so it is half continuous, which is true. Okay, so these fragments which are produced are the one that we call Okazaki fragments. They were named after a certain scientist who discovered this process. Okay, so they are called Okazaki. Okay, so talking about the spelling, Okazaki fragments. That's the spelling. Okay, fragments. So these are Okazaki fragments. All right. So and then I want to talk about another enzyme here, which is very important. So as the replication is occurring, you've seen that we've got these two strands. It is forming a fork, which we call replication fork. So to prevent this replication fork, or these two strands from snapping back together, there's an enzyme called SSB. What does it mean? SSB. So SSB, just an enzyme which means, or SSB stands for the word single-stranded binding proteins. So SSB just means single-stranded what? Binding proteins. All right. So single-strand binding proteins is what we call SSB. So this enzyme prevents the snapping together of these two strands. Okay. So to prevent them from coming back and snap together, there's an enzyme called SSB which prevents that. All right. And then as the replication is ongoing, because we know that. Okay, so as the replication is ongoing, there's going to be torsion force. So torsion force is a force which is created when you twist something. All right, because when you twist something, you find that when you let it go, it will start rotating. Okay, the string will start rotating like that. All right, about the axis. All right, so now this is what we call torsion force. So to prevent torsion force and the damage in the DNA when you are doing replication, there is an enzyme which comes in, which is going to bind there. It binds there. All right. So this enzyme, it is called DNA gyrase. DNA gyrase. So DNA gyrase is just a type of topoisomerase enzymes. Okay. So DNA gyrase is one of the type of topoisomerase enzymes. So it prevents the torsion, or it minimizes the torsion force. Okay, which is created in the DNA strand as it is replicating. All right. So we said that here we've got Okazaki fragments. So short sequences. And then this is the primer. And another short sequence there. And the primer, sorry. So this is the primer, then this is the primer, okay, which is a short sequence of RNA. Then this side, you've got a continuous strand, all right? So this strand, this one, which produces a continuous strand, it is called the leading strand. So this is the leading strand. So we've got the leading strand, then this one which produces the discontinuous, or which produces the Okazaki fragments, it is called lagging strand. So the lagging strand, it is this one. Okay? So DNA polymerase 1, what does it do? So DNA polymerase 1 is going to remove the primer. Okay? So it removes the primer from the copy of the DNA or from the Okazaki fragments. So that's what it does. So it's going to remove the primer on the lagging strand, because the Okazaki fragments, remember, they are produced on the lagging strand. Okay? And the lagging strand produces a discontinuous copy of the DNA, while the leading strand produces a continuous copy of the DNA. So the leading strand, we know it is this one, and this one is a lagging strand. Okay, so DNA polymerase 1 is going to remove the primer from the copy which has been produced, or from the Okazaki fragments, because the primer will remain attached to the Okazaki fragments. Alright, then afterward that, there's going to come another enzyme which is called DNA ligase. Okay, so what does it do? What is the function of DNA ligase? So DNA ligase, it is going to form a bond. Alright, so a bond between these two Okazaki fragments. Or we can simply say it seals the gaps, okay, or the necks between the Okazaki fragments. So it's going to combine these two Okazaki fragments to form one copy or one strand. Alright, so it's going to make a complete strand. So those gaps will be sealed. So this gap will be sealed, another gap will be sealed, all the gaps will be sealed, and it's going to be a long strand, one long strand. Okay, so it's going to be a complete strand, because we've got this continuous strand, and then here we've got a discontinuous strand. So when these necks are sealed, it's going to be a complete strand. So DNA ligase, it seals the neck of the Okazaki fragments, while DNA polymerase 1, the function is to remove the primers, alright, from the copy or from the Okazaki fragments. Alright, so these enzymes, they are very important and you need to know them. Okay, so we said ligase seals the neck. So ligase seals the neck. Okay, then here DNA polymerase 1, DNA poly 1, it's the one that removes the primers. Alright, so now let's talk about we can talk about the exonuclease activity of DNA polymerase 3 and DNA polymerase 1. Okay, so we can say exonuclease activity, exonuclease activity. So exonuclease activity, it is activity, or it's a process which involves the pairing of a DNA molecule or a copy of the DNA molecule, and also proofreading the copy of the DNA molecule. Okay, so as the DNA polymerase 3, as it is moving from 3' to 5', producing the DNA copy in the 5' to 3' direction, sometimes it makes mistakes. Okay, sometimes it makes mistakes. So what happens when it makes a mistake? The DNA polymerase 3, it immediately go back and then correct that mistake. Then that's when it will continue. So those mistakes can be mismatch, okay, whereby a different base is paired with another base. Okay, for example, let's say that H9 is paired with guanine instead of thymine. So that is mismatch. So what it's going to do, this DNA polymerase 3 will go back. So it's going to move in a 3' to 5'. Sorry, in a 5' to 3' direction. Okay, so it's going to go back and remove the what? The mismatch or the error which has been created along the way. So that's what we call proofreading. Okay, so that's what we call proofreading. So it's very important. Then this exonuclease activity, for exonuclease activity for DNA polymerase 1. DNA polymerase 1 it is 3' to 5' and 5' to 3'. It is able to move in either ways. Okay, but DNA polymerase 3, it only moves in one direction. All right, but this one is able to move in other ways. So for example, if it makes a mistake, it's going to go back and remove everything and then continue. All right, so it is able to go back and continue again. So it is in both both directions. All right, so it is in both directions. But for the DNA polymerase 3, it is 3' to 5'. For this one, it is both directions. So DNA polymerase 1, it is most used for repair. Then DNA polymerase 3, it is used for proofreading. So you need to know these 2 points. Okay, so the exonuclease activity of DNA polymerase 1 is for repair. All right. Then for DNA polymerase 3, it is for proofreading. So guys, this is all I got. Make sure that you subscribe to my YouTube channel if you've enjoyed the video, so that you support my work, so that I'll be able to make better videos, guys. Thank you very much for watching this video. Thank you.